Hi, good evening. This is Anna Hatfield, and this is part 2 of our lab assignment for week 2 for Bio225. This is the part where we get to talk about the bacterial enumeration of serial dissolutions and plate counts. The serial dissolutions and plate counts, that would be the calculation number of colonies on the plate, and we did 6 of them. It took about almost 48 hours, total of 48 hours of incubation for them. I went ahead and prepared, just like I did for the broth. I did all of them in one night, and I let them sit for 12 hours, which was the agar solution. Afterwards, I went ahead and added the Cerevisiae yeast to every single dish. But what I did in my last video was I took from my first vial and I took 0.25 ml from the first I forget what they're called a vial, and I had a total of 6 vials, and I went ahead and repeated that, and it slowly but surely, of course, was weakened by the time it got to vial 6, because you started to slowly dilute the yeast. Anyway, so I have my my dishes here, and I'm going to start with 1. Now 1 is very interesting because we had to put 4 drops in each slide or dish, and it is not a viable result. I cannot see colonies. There's too many to count. It is very busy, so we would consider that non-viable, I believe. Okay, so that was number 1, and this is number 2. This one's actually pretty neat as well. I can count the colonies on 2. There appears to be about 30 to 40. So the count for plates 25 to 250 colonies are considered to be viable. That was 2. For 3, I can see also the colonies there. It appears to have about 30. You can see them there. So looking at my book, 54 times 100 would be 54,000 bacterial. If it had 54 colonies, and then you would times that to 1,000, so that would be 54,000 count. So this is definitely probably around there. That was 3, and this is 4. You can count literally like 10, and they're all together and up here. So yeah, this probably would not be a good result to go by if I was the FDA. This one is very busy. It has many colonies there. So this plate would be, I guess, what they would say also not viable. And then my number 6 is extremely I can't even, I don't even know what that is. It's very just smudged, and you can see just big giant circular dots. So it didn't really spread into the tiny colonies like this one here. And those were my results for my for the bacterial enumeration dilutions plate counts. Thank you.